and damage. Dr. Kumar.
Good morning, everybody, and thank you, Mr. Chairman. And I would also like to thank the organizers for inviting me here to share some of my results on radiation protection by gamma tocotrienols. The topic of my presentation today is a question whether tocotrienols protect against radiation sickness and damage. And the next 20-25 minutes, I will present data, and at the end of that, you yourself should decide whether it can protect as a function as a radioprotectant. Radiation protection is necessary for several radiation exposure scenarios like nuclear accidents, terrorist event, extended space travel, therapeutic radiation exposure, and in all these cases, it is necessary for the first responders to go into a radiation field and conduct rescue operations. In such operations, the first responders who are going there needs to be protected, and for that also you need radiation protective agents. Radiation protection can be done either giving the protectant before radiation, so that, as in the case of first responders, when they go into a field, they can be well protected. And it can also be given to mitigate or treat the radiation damage after exposure, after properly assessing the radiation dose to which the individual is exposed. Then you can give the proper dose of the mitigating agent. It is also useful in attenuating the damage in a combined scenario situation. That is, when radiation is combined with biological agent or when radiation is combined with chemical agent, there is an enhanced damage than with radiation alone. So if you reduce the damage by the radiation component alone, then you will be able to reduce the total damage by the combined scenario situations. So in order to develop radioprotectors in our institute, in my ex-institute, I retired from there last year, we have been conducting studies with the nutraceutical agents. Particularly, we have been using tocols. And among the tocols, what we found was the tocotrienols have a better protective profile than alpha tocopherol. So the rationale for selecting gamma tocotrienol is that it has the highest activity against oxidative damage among tocols, and also not only that, it also inhibits peroxynitrite. As you know, peroxynitrite is a product of superoxide and nitric oxide. And this product, peroxynitrite, is more potent as a toxic agent than either one of superoxide or the nitric oxide. So in order to test this gamma tocotrienol, the experiment, this next couple of slides compares the antioxidant effects of alpha tocopherol and gamma tocotrienol. As you can see, the gamma tocotrienol has a better antioxidant activity than alpha tocopherol. It's an in vitro system for testing the lipid peroxidation activity. And if you compare the tocotrienols, particularly gamma tocotrienol and delta tocotrienol, then you can see there is actually no difference in the antioxidant activities of these tocotrienols. And in order to test the inhibition of peroxynitrite, what we did was we exposed the animals to radiation and isolated the aortic rings from them and looked at the induction of peroxynitrite formation. Here you can see three panels. The first panel shows the peroxynitrite formation immediately after radiation. Next panel 4 hours after radiation, and the last panel shows 3.5 days after radiation. You can see that at 4 hours after radiation and 3.5 days after radiation, there is a significant increase in the peroxynitrate as compared to the GT3 treated animals. And in all the three panels, you can see GT3 inhibits the peroxynitrite formation, indicating that it's an also inhibitor of radiation-induced nitrocytic species. Now, having known the antioxidant and the peroxynitrate activity, we developed the experimental protocol for studying the radioprotective properties of GT3. And in this protocol, the mouse are given gamma-tocotrienol 20 to 24 hours before radiation. And the radiation damage in these animals are monitored by three factors. First, hematology, then gastrointestinal factors, and ultimately, the ultimate test of radiation protection is whether the drug can prevent the lethality in the animals. So let us look at the hematological aspects. When you isolate the bone marrow, stain the bone marrow immediately after radiation, this is the pattern that you see. But 2 days after radiation, you can see there is already a difference being developed in the vehicle-treated animals, that is, the animals which did not receive GT3 and the animals that received GT3. Here you can see there are still some white cells, but in this case, it's mostly dominated by the hemolyzing red cells. If you go to 7 days after radiation, you can clearly see the difference between the vehicle-treated animals and the gamma-tocotrienol-treated animals. The gamma-tocotrienol-treated animals tend to be approaching the normal animal's bone marrow. And 13 days after radiation, the vehicle-treated animals, the bone marrow is completely denuded, and the GT3-treated animals are protected from damage of the bone marrow. But just by looking at the stain itself is not enough. So we proceeded further to see how the stem cells and the progenitor cells in the bone marrow can be protected by the gamma-tocotrienol. And the experimental protocol for that is to take out the bone marrow or even the cells from the peripheral blood, and then subject that to lineage negative population, sort out the lineage negative population, and that lineage negative population then marked for hematopoietic progenitor cells and stromal progenitor cells and the endothelial progenitor cells. And you can see that in all the three, this is the experimental model that we have used for the identification of the various progenitor cells. In the case of hematopoietic progenitor cells, we have looked at the pattern 24 hours, 48 hours, and 72 hours after radiation exposure. See in the 48 hours and 72 hours, gamma-tocotrienol and gamma-tocotrienol plus AMD. AMD is a drug which is injected to the animal 1 hour before the removal of the blood, so that whatever hematopoietic progenitor cells are in the bone marrow, they are mobilized in the circulation and they can be isolated and identified. So those animals which has received GT3 and AMD are significantly higher numbers of HPCs at 48 hours and 72 hours after radiation. And if the pattern is almost identical when you go look at the endothelial progenitor cells. Here again you can see one of the difference between endothelial progenitor cells and the hematopoietic progenitor cells is that even at 24 hours, you can see the significant difference between the control-treated animals and, or the vehicle-treated animals and the naive animals with the EPCs or endothelial progenitor cells. The story is almost identical for the stromal progenitor cells as well, except that in the stromal progenitor cells, the maximal effect is in 48 hours. So in the HPCs, the maximum effect is 48 and 72 hours. At the EPCs, it is at 24 hours, and the SPCs, it is 24 and 48 hours. Now, these are only the numbers of the various progenitor cells. How do we know they are functionally real progenitor cells? In order to do that, we had to look at their ability to form the colonies, which are shown in the next 3 slides. In this, the peripheral mononuclear cells are treated with specific colony-forming units. For example, in this case, with SPC colony-forming units or the EPC colony-forming units. And you can see there are clear formation of the colonies in both cases, indicating that they are not just simply numbers, but they are functionally working progenitor cells. And when you count the colonies, you can see then almost an identical pattern as in the case of number of the various progenitor cells. And again, you can see 48 and 72 hours after radiation. SPC colonies were significantly more than the vehicle-treated animals. Similarly, endothelial progenitor cell colonies as well, and the stromal progenitor cells also. So, looking at the bone marrow and the peripheral blood, it is quite clear that gamma-tocotrienol definitely protects the progenitor cells as well as the formation in the, not only the number of progenitor cells, but functionally active progenitor cells in the mice. So it is not only the bone marrow that is protected, but if you look at the extramedullary hematopoiesis, for example, in spleen, what we have shown is the spleen harvested 12 days after radiation. You can see the vehicle-treated animals, the spleen has become much smaller as in case of radiation. But when the GT3 is also given to those animals, the spleen returns to almost the normal level. But it's not only the size of the spleen, but the various colony-forming units in the spleen can also be identified by staining the spleen with Bowen solution. Here you can see the vehicle-treated animals have only very few colonies, while in the case of GT3-treated animals, several colonies can be seen, and you can see in the figure, vehicle-treated animals have barely 5 colonies, while the GT3-treated animals have almost 100 colonies or more. So whether it is the extramedullary hematopoiesis or the bone marrow hematopoiesis, GT3 has a clear protective action against radiation-induced hematological dysfunctions. Now let us look at the gastrointestinal tract. If you expose the animals to various radiation doses ranging from 2 to 16 gray, if you look at the radiation doses from 9 to 15 gray, you can see a distinct difference between the tocotrienol treated animals and the vehicle treated animals. These are the vehicle treated animals and these are the tocotrienol treated animals. The surviving fraction of the clip cells in these animals are, the animals treated with gamma tocotrienol are much greater as compared to the vehicle-treated animals. If you look at the stain of the cross-section of the intestine, In the normal animals, vehicle treated without radiation, you can see the crypt cells, very well formed crypt cells. But when they are irradiated at 11 gray, you can see the dissemination of the crypt cells by radiation. And when they are treated with gamma tocotrienol, the crypt cells regenerate in the GT3 treated animals. It is not only the crypt cells, but also the apoptosis induced by radiation in these animals are also reduced by GT3. In the first panel is an unirradiated animal where you can see very barely one or two apoptotic bodies, while in the irradiated animals there's quite a number of apoptotic bodies, and when they are treated with gamma tocotrienol, the number of apoptotic bodies are considerably reduced. In addition to the crypt cell dissemination and the apoptosis, another way of looking at the GI damage is by looking at the recovery of the enterocyte mass as well as the reduction of bacterial translocation. Bacterial translocation is that when the intestinal wall is damaged, the several bacteria which is present in the intestine, they translocate into the blood, and from the blood it colonizes several tissues, resulting in the death of the animal. Here you can see when the enterocyte mass can be assessed by looking at the plasma citrulline level. In the case of vehicle-treated animals, 7 days after radiation, there is considerable reduction in the enterocyte mass, but when GT3 is given, that is returned almost back to the normal level. If you look at the bacterial DNA in liver, which is an indication of the bacterial translocation, the vehicle-treated animals have a large amount of bacterial DNA in the liver, while GT3 treated animals have considerably less DNA in the bacterial DNA in liver. So these two studies, hematological studies and the gastrointestinal studies, shows that GT3 is a good radioprotectant. But the ultimate test for a radioprotectant is to see whether they can protect the lethality of the animals. And that is probably the next slide here. Here, what Dr. Ghosh and her colleagues who was working in one of my projects was done was that they treated the animals with various doses of gamma tocotrienol, varying from 50 to 400 milligram per kilogram body weight, and compared that with the vehicle-treated animals. Here you can see, I need some water. You can see that even 16 days after radiation, all the animals treated with the vehicle have died. But even the lowest dose of gamma tocotrienol given, which is 50 milligram, there is a significant difference in the protection by after radiation. And 100 milligram and 200 milligram, which are the two topmost lines, there is almost 100% protection against radiation damage. Somehow, when 400 milligram per kilogram body weight of GT3 was given, there was more lethality in those animals than the 200 and 100 milligram per kilogram body weight, indicating that probably when you reach a higher concentration, there may be some interaction between radiation and gamma tocotrienol, which causes a toxic effect. So, What are the general conclusions? The conclusion is GT3 is a non-toxic, inexpensive, and stable radioprotectant. Protection by GT3 may be associated with a superior ability to inhibit oxidative and nitrosative stress. GT3 can protect from radiation-induced bone marrow and GI as indicated by the protection of HPCs, EPCs, and SPCs, and GI crystals. Now you decide whether we started out with the question, do tocotrienols protect against radiation sickness and damage? And I told in the beginning that radiation protection, damage and sickness is characterized by hematological symptoms and gastrointestinal symptoms, and finally by the lethality. And we have seen that all these three factors, monitoring factors, were protected by GT3. And so I think I don't need an answer from you. We can safely assume that GT3 is a fairly good radiation protection agent. I would like to look at the future directions. Where do we go from here? First of all, we have to develop an orally effective radioprotectant. In all these cases, the GT3 was given. GT3 was given after as a suspension and given as a subcutaneous injection. But in case of a mass casualty situation, giving a subcutaneous injection may not be practical, and so we need to develop orally effective protectants. And then we have to conduct toxicity and efficacy studies in large animals in case we proceed further with this so that ultimately it can be used in humans. We have to do the toxicity and efficacy in large animals. Unfortunately, because radiation is a damaging has damaging effect, we cannot use humans as volunteers for this testing the drug, as in the case of the drug development process for other diseases, because the diseases are already existing. In case of radiation damage, it is not existing there. So we had to depend on large animal studies. And then, of course, initiate pharmacokinetics and pharmacodynamic studies with gamma tocotrienol. Of course, we have some data, but when you go to approach a regulatory agency like the Food and Drug Administration in the US, we had to have conducted that in what is known as GLP practices. I would like to acknowledge the US Defense Threat Reduction Agency for the financial support. Thank you. If you have questions.
Thank you very much.